Okay, so the same thing when we express in Punnett square, okay, the same thing when we express in Punnett square method, okay. Suppose this is capital T and this is small t, okay. During gamete formation there will be segregation of this pair of allele, okay, pair of gene. So what happens is, this is tall, this is dwarf. So if these two fertilizes, then it will be capital T small t, these two capital T small t, again this and this. So all in F1 generation, all the plant will be tall, though it is heterozygous, but all will be tall, okay. The same monohybrid cross which Mendel did, only expressing in Punnett square method, okay, F1 generation. Now the same F1 generation, if F1 generation it is allowed to undergo self pollination, then so if this gamete fertilizes this gamete, it will be capital T, and this two capital T small t, okay, this two capital T small t, and this two will be small t. So this is F2 generation. In F2 generation, though it is, the ratio will be 3:1, because 3 of the plant will be tall and 1 will be dwarf. But out of this 3, 1 is homozygous tall, 2 are heterozygous tall, 1 is homozygous dwarf, okay. So one thing, so in F2 generation, phenotypic ratio means the, when we look into this plant externally, what we see, okay, that will be phenotypic ratio. The externally, when we look into this plant, we will see that 3 plant will be tall, okay, in height, and 1 will be dwarf externally. So the phenotypic ratio will be 3:1. But the genotypic ratio, genotypic means not only externally how it looks like, but when we analyze it internally, okay, whether it is homozygous or heterozygous tall, then that will come under genotypic ratio. So in this genotypic ratio, we got 1:2:1, because 1 was homozygous tall, 2 were heterozygous tall, and 1 was homozygous dwarf, okay. So the genotypic ratio will be 1:2:1. So this is all about monohybrid cross which Mendel did, okay, just expressing in Punnett square. Now monohybrid cross means only one character is taken into consideration. He observed only one character, he focused in only one character of the pea plant, that was height. That's why it is called monohybrid cross, okay. Single character is taken into consideration. Now again what he did was, he did a test cross, he did again a test cross. Now in this test cross, he allowed this F1 generation, that is capital T small t, okay, this progeny, he allowed this F1 generation to cross with one of the parent, which was recessive, okay, dwarf, recessive parent. So during gamete formation, these two segregate, okay, they separate, again these two. Now in this cross, he got capital T small t. If these two, then it will be small t, and if these two, it will be small t. So to his surprise, in the test cross, he got 2 tall plant, though heterozygous, and 2 dwarf plant. So the test cross result in the ratio 1:1. So this is the test cross in which F1 generation, okay, F1 progeny was crossed with one of the recessive parent, okay, and he got the ratio 1:1, okay. Mendel got the ratio 1:1. So this is all about test cross, and this is all about monohybrid cross which Mendel did, okay, in his experiment. Thank you, class.